Hello everyone, and welcome to this podcast for genetics. In this podcast, we're going to talk about genomics. This is really perfectly timed at the end of the course to talk about genomics, because genomics is kind of bringing everything together that we've learned. Remember, as the semester has gone on, we talked about how nucleotides came together and formed a double helix, the DNA molecule. And we talked about how on this DNA molecule there were certain genes, and how this gene could be transcribed into an RNA molecule. And then we talked about how this RNA molecule could be translated into a protein of various shapes and sizes. We also talked about how this whole process here from the gene to the finished protein was highly regulated. And this was important because if you have an individual here, doesn't have to be a human, but could be a human, they have many different cells, and those cells in different tissues have to turn on the right set of genes and not turn on other genes. So this regulation was really important. We talked about how this individual here with another individual could produce progeny by making gametes through the process of meiosis, and how this individual here looked a lot like these two parents, but might also have some new traits. We talked about all the laws of inheritance and the variations of those laws that explained how we see certain traits. But now what we want to do is we want to take our knowledge of genetics, the process of how DNA turns on genes, how it copies itself, how it's passed on to our children. We want to take that knowledge to try to understand the whole organism. How do we learn more about individuals? The organism. How do we know more about this organism here, this stick figure human I drew in purple here? How do we know more about him or her? How do we know more about this other human stick figure I drew? How do we know more about their progeny? With that in mind, how do we learn more about a group of individuals or an entire community? How do we learn more, using our understanding of genetics, how do we learn more about this group of individuals? Or how this group of individuals interacts with other individuals of different species in this larger community? And finally, what can we learn about evolution from our general knowledge of genetics? So I'm going to erase this to give myself a little bit more room. So we consider evolution here with our three domains of life, of bacteria, archaea, and eukaryotes. And of course, considering the four kingdoms under eukaryotes: animals, plants, fungi, and protozoa. How can we take this knowledge of genetics and help us understand the evolutionary relationship between these different kingdoms, between the species within these kingdoms, between these different domains? We want to understand how organisms in eukaryotes compare at an evolutionary level with archaea and bacteria. And as you might have guessed, to answer these questions, we need to understand genomics. So let's provide a brief definition of genomics. And I took this straight from your textbook's glossary. It's the study of the content, organization, and function of genetic information of the whole genome. Now, to get to the point where we actually have all this information, it took a long time to get there. But in order to get here, we needed a map, just like you might need a map to get to anywhere you want to go. And we've talked about at least one of these maps before. We've talked about genetic maps. If you remember, a genetic map was made by understanding the relationship of genes based upon their recombination frequency. So if at this locus we had the alleles gene big A and little a, and at this locus we had the alleles big B and little b, we could determine what we called map units by observing recombination frequencies. So the number of times that we had a crossing over event here told us a general relationship between the distance between these two genes. So if there was a 20% recombination frequency, we would say they were 20 map units apart from each other. So this is useful information because it does give us this general information about the order of genes. However, there are some problems with this. The first, it provides genetic maps provide only a rough approximation of the distance between genes. And what we've learned is that when we finally have understood the actual nucleotide sequences between these two genes, that the genetic maps weren't always as accurate as we thought. They're pretty good, but not as accurate as we might have hoped. The other problem is that genetic maps are only as good as the genes you are aware of. For instance, there may be a gene C here, and maybe for whatever reason we never knew about this gene C, and so we never picked it up. Or, more importantly, remember that most of our genomes in humans and in other organisms is made up of this non-coding DNA. The genetic maps tell us nothing about this non-coding DNA because it only provides us information with genes that have a phenotype. If there's no phenotype associated with regions down here or over here or between genes, we can't really get a good estimate on their location or the distances they are between other parts of the chromosome. After genetic maps, the next thing that came along were things called physical maps. I'm not going to go into great detail on physical maps, but I want to give a general idea of how they were used and what kind of information we got from them. So you may have a long piece of DNA here, but you can't really see it. But what you can do is you can put it in a tube, and in this tube we can add restriction enzymes. Restriction enzymes, remember, are sequence specific. So any given restriction enzyme may cut this long piece of DNA at different places. And maybe you put in other restriction enzymes that cut it at even different locations. And through a long and arduous process, as you're chopping the DNA up, you're getting different fragments of different sizes. And you begin to compare the sizes and where they have overlaps. And ultimately what happens is, from this tube of DNA with restriction enzymes, we're able to piece things together because of these overlaps. So these regions here are overlapping. So you may cut it here with the red restriction enzyme, and then maybe with this brown restriction enzyme you cut it there. And so now you have this little bit of an overlap between those. And based on that, you can start to begin to get an idea of how this whole chromosome pieces together. However, if we really want to understand the genome, mapping the chromosomes together is helpful. What we really need is DNA sequence. What is the actual nucleotide sequence of this? Then we can start to understand the different genes that are located here. We can start to understand more about the nature of those genes. So how do we go about doing that? Well, we could take, say, this here sequence that's been cut, and we can put it into a plasmid, and we can sequence this. That's not too much of a problem. We could sequence this. And we could do that with all the fragments. So it seems like it's a doable process, but there were some obstacles. And I want to talk about some of the obstacles that early genomic investigators faced trying to get this DNA sequence. So some obstacles to sequencing genomes. The first is that genomes are huge. They can be anywhere from, say, 1 million, maybe some viral genomes are less than 1 million, but usually between 1 million and billions of base pairs. And when we sequence genomes, kind of like as I mentioned on the last slide, by cutting genomes up and placing them in plasmids and sequencing them that way, as I said, that's pretty simple to do. But the problem with that is, when we sequence that way, we can only sequence, say, between 500 to 700 base pairs at a time. And in the early days of DNA sequencing, it was even less than that. So if you're sequencing over 1 billion base pairs and you're only doing it at 500 to 700 base pairs at a time, it's going to take a lot of time, money, and effort to get that done. So with that in mind, to sequence something like the human genome, which is 3.2 billion base pairs, it's going to take an immense amount of time. Another problem, we can't sequence one end of the chromosome to the other end. If we have our chromosome here, we can't just begin sequencing here and collect data all the way till the end. It just doesn't work that way. There are regions of the chromosome that, for a variety of reasons, are difficult to sequence. So you really did just have to chop it up, as I described up here, and sequence small little chunks of this genome at a time, and then try to piece them together. And so another obstacle to this whole process was technology. Now, how these genomes are actually sequenced, the specifics of it, the technology behind it, we're not going to get into that for the purpose of this class. You can read more about it in your books, and if you want to, we can talk more about it as well. But for this class, we don't need to know it. But you should know that through a lot of hard work, dedication, money, and an increase in technology allowed us to begin to sequence whole genomes. And now we can even sequence multiple genomes at the same time, which we'll talk about in a moment. What I'd like to do now is to list some of the genomes that have been sequenced. And with some of them, I want to explain why their particular genomes are considered landmark discoveries. So the first genome to be sequenced was bacteriophage. It was sequenced in 1982. I'm not going to ask you years on an exam, but just putting that there for your reference. It was pretty small, only about 49,000 base pairs. But the amount of time it took them to sequence these 49,000 base pairs, we can now sequence base pairs that are millions of base pairs in that same amount of time, less than that amount of time. Another landmark genome that was sequenced was Haemophilus influenza. And this was sequenced in 1995. And the reason why this was important was because it was the first free-living organism. It's a kind of a bacteria. It's about 1.8 million base pairs. The third one to be sequenced is one that's near and dear to my heart, and that is Saccharomyces cerevisiae. The common yeast is Saccharomyces cerevisiae. This was an important landmark genome that was sequenced because it was the first eukaryote. And this was done completed anyhow, in 1996. And it contains about 12 million base pairs. The next important one to be sequenced was E. coli. Now, E. coli is another bacterium like the Haemophilus influenza. It just came a little bit after Saccharomyces. And it was sequenced in 1997. And it had about 4.9 million base pairs. This was an important one because E. coli was, and continues to be, an important model organism. Up to this point, much of what we had learned about the molecular genetics of DNA was learned using E. coli. The next one was a very important one, and this is C. elegans, the microscopic worm that you have worked with in the lab. It was sequenced in 1998. It has approximately 100 million base pairs. And the reason this was of such importance is that it was the first multicellular organism to be sequenced. In fact, much of what they learned about sequencing the C. elegans genome, they used to really speed up the sequencing of the human genome. So really, without C. elegans being sequenced, it would have taken a lot longer for the human genome to be sequenced. The sixth one here I want to mention is, shortly after C. elegans, Drosophila melanogaster was sequenced, and that was done in 2000. It was 132 million base pairs. Now, it wasn't the first multicellular organism sequence, but it was an important one for the same reason that E. coli was. Fruit flies have historically been a very important and continue to be an important model organism to understand genetics. So this was an important one to get sequenced. And the last one to put down here is one that you may have heard of, humans. Our genomes were sequenced, the rough draft in here was in 2000, and then they got finalized in 2003. Our genome, as I mentioned before, has a little over 3 billion base pairs. So that's huge compared to these other ones. Now that we have all of these genomes and their sequences available to us, we can start to try to understand each of these organisms. And we can start to try to understand the evolutionary relatedness of all of these organisms. We can start to look at humans and our genome and try to understand our development, our evolutionary history, various diseases that afflict humans. This really opens up a lot of opportunities, but also, as we'll learn, a lot of responsibilities. So what is the human genome? Let's spend a little bit of time talking about that. What is the human genome? To understand this question of what is the human genome, it's important for us to understand where the genome came from that was sequenced. Remember, on this planet, we have billions of people, and we weren't the first people here. There have been people on this earth for a very long time. And so what we've really done is taken a few individuals and sequence their genomes. And so when we talk about the human genome, we're talking about a relatively few number of individuals. How many, it's not real clear. But they took a couple people from Buffalo, for instance, and a few other individuals. And when they started sequencing the genome, they used those few individuals to represent the entire human genome. Small problem with that is the fact that really on this earth, we likely have a billion or more different human genomes that differ slightly in some instances and maybe largely in other instances. So we have come to call this human genome coming from these relatively few number of individuals as a reference genome. This is true with genomes sequenced in other organisms as well, but we're just talking about humans now. So a reference genome. Doesn't mean it's the most common genome. It doesn't mean it reflects every human being on this earth currently or in the past. It means it's a reference for us humans. And in fact, when we get better information by sequencing other individuals, we change what we have recorded as the human genome. It's constantly being updated. So the question is, how confident are we that this reference genome taken from a few individuals really represents or is a meaningful representation of the entire human population? We can be pretty confident of that. And the reason for that is that the difference between any two humans, say a human in Cleveland, Ohio, and a human in Shanghai, the difference between these two individuals from opposite ends of the world is minute. In fact, if you compared any two humans on Earth, they would be about 99.9% genetically identical. That 0.1% is what helps account for the vast diversity we see within our population, but it's still only 0.1%. It's a very small number. If you consider the fact that, I should put here down here, between humans, any given humans, I wanted to make that distinction because I also want to remind you that between a human and a chimpanzee, it's about 96% genetically identical. So this is humans to chimps. We're still pretty similar, and the reason for that, I think this is a very important thing to keep in mind, so I want to make sure I write it up here, is that humans are a young species. Our species has not been on this earth very long. And because of that, that's why the difference between any two humans is so small. We often make a big deal about that small difference, but in reality, not much difference. So with that in mind, these few individuals are a good reference for the entire human population. Not perfect, but still good. Okay, the next thing I want to talk about is this field called bioinformatics. You know a little bit about this because of some of the activities we did in lab. But what we used in lab was kind of the end product of what bioinformaticists do. I first want to tell you what a bioinformaticist does, or what it is. This is an interdisciplinary field that combines molecular biology, genetics, so to speak, and computer science. Now this field boomed as genomes were being sequenced. Because of all the genomes being sequenced, there was a lot of data. So the field of bioinformatics helped store, catalog, and analyze these large sequencing data. Also, what they did was that they made this data easily usable. So someone like myself, who doesn't have a really strong background in computer science, but I can go to websites and obtain data easily about genes I'm studying. What maybe took a molecular biologist years to accomplish, we can now accomplish in an afternoon because of what bioinformaticists have done to help this field of genomics. Stemming from genomics, the sequencing and understanding of an individual's genomes has been this relatively new field called metagenomics. Now, metagenomics is the sequencing genomes of whole communities. So to remind you, a community includes all of the different species within a particular area. So because of the advances in sequencing tools, bioinformaticists, and other technological advances, we can now sequence these whole communities, which is really fascinating. Now, it's not perfect, and there are still some limitations to it, but it has been very useful in studying microbial populations. So useful in studying microbial populations. This has been useful for two particular reasons. One is we can now study unculturable microbes. Now, traditionally, what a microbiologist would have done was they would have taken a petri dish shown here and have taken a sample from the environment and have plated it on here. And then what happens is that various colonies form. And these colonies may be different species, they may be the same species, but they grew. And because they grew, they could analyze them. So these were considered culturable. But what we've learned is there were even more colonies present in this sample, we'll make this our sample here, that they brought from the environment. But several of these samples, I'll just make a dot here, weren't able to form a colony. They were not culturable. However, we know that these must be important because they were in the environment, but we couldn't study them. But now we can study them because now what we can do from this entire community from the environment and sequence it. Sequence the DNA from all members of the community. Now we can finally start looking at some of these unculturable microbes because they don't actually have to grow in order for us to get DNA from them. But we can learn a lot now about the entire community that we couldn't before. The second reason is we can now begin to understand microbial communities. And a booming field right now is that is associated with the human microbiome, meaning all the microbes that live in humans, in our intestinal tracts, in our skin, and how these different microbes affect human health and other traits. The list of traits that are affecting humans based upon their microbiome is growing and growing, and is rather impressive what these microbes are doing in our bodies and how they're helping control us in many ways. So we'll talk more about this in class, but this is an excellent plug for you to think about taking a microbiology class later on during your curriculum here. The next thing I want to talk about is something called transcriptomics. This is the study RNA molecules produced from a particular genome. And it's not just studying the RNA molecules produced from a particular genome, it's comparing that set of molecules from one population to another population. So with that in mind, there are many different applications. For instance, if we had a human here, my lovely stick figure human here, if you want to know the genes that are important for the heart versus the genes that are necessary for the liver, you could take RNA samples from both. And what that would tell you are the genes being activated for heart function. And from the liver, it's going to tell you the genes being activated for liver function. And of course, it tells you when it's not being activated as well. They'll compare these and they'll say some of these genes are expressed in both, some are expressed in one or the other, and that gives us a greater understanding of these two organs. We could also compare the genes being expressed in this person's heart with the genes being expressed in this person's heart. So now that we have the collection of RNAs from these two individuals, if one of them, say, suffers from an arrhythmia, an altered heartbeat, what we might be able to do is begin to understand if the gene expression from these two individuals differ. And maybe that will help us understand why this person doesn't have the heart arrhythmia, but this person does. So the kinds of studies are enormous, the different kinds of options you have. You could study between humans and other organisms. And though I'm talking about humans, it should not be confused that we only do this with humans. I study yeast, and this is a very powerful tool in studying yeast. Almost every molecular biologist takes advantage of this very important tool in one way or another. So when researchers are looking at this, what do they see? So in this example, let's look at the RNA from a healthy person versus over here, the RNA from a cancer patient. And let's say it's skin cancer. So they're going to take the RNA from both of these sets of tissues, and they're going to use a technique called microarray. And I don't want you to learn the technical details of this, but I would like you to be able to look at some data and be able to make a prediction. So these arrays contain spots for all of our genes, potentially all of our genes. And if a particular gene is activated, the RNA will hybridize to those different boxes here. And they've used a color scheme to show red and green dots. And so for instance, let's just say that these dots up here were fluorescing green. Different levels of intensities, but that's harder for me to show with a marker. And over here with the cancer patient, these are showing green. And I should also mention that this set of genes are the same set of genes over here. Maybe here we see red, and down here is red as well. And it's a little counterintuitive, but red means the gene is overexpressed. Seems that red to me signifies stop, so I would think it's underexpressed, but that's not the case. It's opposite. Red is overexpressed, whereas green will indicate a gene is underexpressed. Again, it's a little counterintuitive to me, because I think of being expressed, I think of go, and I would think of green as go, but this is the opposite. It is underexpressed. So what we can tell from this made-up data here is that this particular gene is underexpressed. I'm just going to draw a down arrow. Whereas this particular gene is overexpressed with an up arrow. But what we can do now is compare this with the cancer patient. And we can say that these genes here are normally underexpressed, but now they're being overexpressed. And these genes here are normally overexpressed because of the red, but now they're underexpressed. This could lead us to understanding important cancer genes. So this can take us back to some of our understanding of cancer. We know that, for instance, oncogenes in cancers are typically overexpressed, whereas tumor suppressor genes are underexpressed. So this could lead us to identifying important tumor suppressor genes or oncogenes found in cancer patients. The last thing I want to talk about on this podcast is something called proteomics. And I'm not going to say a whole lot about this, but I would like you to know what it is. Just like genomics is the study of the DNA sequence within an organism, and transcriptomics studies the RNA in individuals, proteomics is the study of proteins expressed from a particular genome. Much like the transcriptomics, proteomics will give us a hint as to which genes are actually being expressed in specific tissues. So this plays closely with our understanding of transcriptomics. But that's all I really want you to know about proteomics. Well, that ends this podcast. Make sure if you have any questions at all that you come see me, and I will help answer any questions you might have. This being our last podcast, I don't want to say bye. I want to just remind you that as you move forward with your career here at CMU, please feel free to stop by and chat with me if you have questions about your career, about your schedules, about anything. My door is always open. I also like to say it's been a privilege teaching you this semester. You've all worked very hard, and you've learned a lot. I've been very impressed with the amount of work you've put in, your level of attention, your amazing attitudes, and your strong work ethics. Some of you I've had for the past 2 semesters. Some of you, this is our first semester. It's been a blast teaching you. I hope to see you in another class. But until then, I will see you around.
Sheldon, I can't believe I'm saying this, but I'm gonna miss you.
Please, Penny. As you know, I'm not comfortable with prolonged goodbyes and maudlin displays of emotion, so I prepared a short video. Greetings. As you know, I'm not comfortable with prolonged goodbyes and maudlin displays of emotion, so I prepared this short video. The 4 of you are 3 of my closest friends and 1 treasured acquaintance. Though I cannot state categorically that my life will be diminished by not having you in it, I am comfortable if you choose to believe that. Since you intend to remain in this lawless metropolitan area, statistics suggest that you will succumb to tragic and gruesome ends before we meet again. Live long and prosper.
Sheldon, that's so...
You might want to lock the door behind me. This isn't Bozeman.